Welcome to Space Analog Stories. I'd like to introduce Dr. Reut Abramovich. Reut is an astrobiologist with a bachelor's degree in biology and a PhD in microbiology and immunology. She is a research scientist at the Dead Sea and Arava Science Center in Israel. Former chief scientist and co-founder of DMARS, the Desert Mars Analog Ramon Station, analog astronaut and public speaker. Welcome, Reut.
Thank you, Jess. How's it going?
Oh, it's great. It's great to have you here. Could you maybe start by just giving us a short background of the work you do, how you came up with DMARS, how you co-founded DMARS, and how your love for space started?
Okay, so my love for space was always there as a young child. Anyhow, I started very young, reading science fiction books and watching Star Trek, and I'm a fan of Star Wars and so on and so forth. It's very easy to get hooked by it. Cosmos was the TV show with Carl Sagan, was a big influence on me when I was in 4th grade or something. I remember myself. They had that big episode about, are there life elsewhere in the universe, in the stars above? And I remember literally, I'm standing actually next to the actual window of my childhood room, and I was looking outside through the whole night just to see if something is coming from the sky, things like that. Very romantic view of it. And as time progressed, I got more and more interested, but I also noticed that I'm more interested like on the human aspects, on the medical aspects, on the biological aspects of it, much more than the physical, astrophysical astronomy. And slowly but surely, I found my way into this area again. And I was very, very happy to find it. In Israel, we don't have astrobiology as a, as an undergrad or as a title. But there are a lot of places in the world which do have it. And I chosen one which was lovely, absolutely lovely in Australia. University of New South Wales had the Australian Centre for Astrobiology, and they have, I did my PhD, which is in microbiology and immunology, but like in extreme environments. And I met all these amazing people, Malcolm Walters is like a really well-known geologist, and all the people who know about stromatolites so much, the beginning of life on planet Earth. And I had my own time with stromatolites in Shark Bay, hanging out, and I was part of the Australian Mars Society. So it was a great place to do my master's and my PhD in Australia, and continue with astrobiology, learning about extreme environments, but through this angle of microbiology, how these things work. Yeah.
Sorry to interrupt, but what is stromatolites? That's something I've not heard before.
Stromatolites. So stromatolites is a word. It's combined from two different words, which are originated in Greek. Stroma, which means to spread, and lit from the word litus, which is stone. You can find it in other big words, long words. And stromatolites are very interesting structures within stones, within rocks. They have been discovered about 250 years ago by geologists. But the thing is, the geologists were looking at these rock patterns, and some of these rocks were really, really old. Some were just 10,000 years old rocks, and some were like even billions of years, sometimes 3.7 billion years. And they were looking at this, and they're saying, look, this pattern that we see here, it's like lines, it's conical shapes, it's repeating. We don't think that's physical, just physical. We think there has been some sort of a biological action that made this shape within the rock. So, lo and behold, they found stromatolites, and stromatolites, today we find them. We find these structures of microbial communities that actually build stones. And Australia has a great natural reserve in Western Australia called Shark Bay, where you can see these processes. So what happens is that part of the area you walk in, it's a shallow bay, it's a wonderful beach, it's a great tourist attraction, there's lots of dolphins and sharks and whatnot. Australia, there has to be a shark somewhere, it's Australia. And you can see these rocks, and it looks like little boulders, basically. You look and they have all these boulders, and it looks completely like a rock. But then you go inwards, and in some places where I did my research, when you take a hammer and you actually, smash a little bit of that rock, what you find inside, you find like this living microbial community that has this really hard shell on top. And what you understand is that if you take this process of microbial communities building a rock, but they're doing it for thousands and millions and hundreds, eventually what you're going to have a rock. You're not going to have microbial mats, but you're going to have eventually this rock, and you're going to have to see these shapes. So the research took on, and people are looking for like biomarkers in rocky stromatolites, in really old rocks. They're trying to look for lipids, you know, things that are in the microbial envelope, to see if you can find these biomarkers in rocks. And sometimes they find them, sometimes they're not. They're looking for stromatolites on other planets, on Mars, for instance. Perhaps maybe Jezero crater will have these evidence of stromatolites, because if we find evidence of stromatolites here on Earth, like 3.7 billion years ago, then we think now that at that time, Mars was actually had an atmosphere, had, you know, oceans. So if Earth had life at that time, we sort of think that perhaps Mars might have had as well. And if it was stromatolites on Earth, perhaps you can find something similar. It's not going to be the same thing, obviously, different physical conditions and everything, but still that period of time when they had an atmosphere, it was raining, there was an ocean, there was liquid water, then perhaps you had stromatolite as well. So stromatolites are awesome. You can find them also in lakes, in Canada, some places, I think in China. And it's a fascinating thing because it's the basic of geobiology. Like you can see, and in Shark Bay specifically, so the shallow bay is really, it's really, really long. And some parts of it, when you walk, and I did this, when you walk into this ocean, you're not walking on seabed, you're actually walking on meters of stromatolites, which are building their layer. So you're not stepping on something really hard, your actually foot goes all the way in. It's like a muddy feeling. And then you pick it up and you look and you see these different layers and everything. And it's awesome. It's just an amazing area of research. I haven't done that area for a while. I did it in my PhD. I was looking at nitrogen fixation, just putting it out there. I was looking at nitrogen fixation from the atmosphere in extreme environments, yes. And so that's the whole story about stromatolites. So it will be very exciting to find them in Jezero crater or anywhere else on Mars. We'll see.
Yeah, I mean, that sounds fascinating. I'll definitely talk more about the astrobiology research. But could you also just tell us a little bit what led you to co-found the DMARS research station in Israel?
So the same thing that led me to co-found the Israeli Mars Society, I sort of noticed there was a lack, and there's a lot of interest going on in Israel, in the world, what's happening with Mars and everything. So if you really want to look at the big overview, then Mars is the Israeli Mars chapter is for everyone and everyone, everything, every angle you want to put onto the Mars topic. Arts, architecture, biology, science, rockets, SpaceX, Elon Musk, aliens, how you want to do it. We have the Facebook, group for it, and we're happy about it. But DMARS is a different animal. So DMARS is like making this a reality in the most real way that you can have. And I was very lucky, to join these people who had the same vision as I had. And they were saying, you know what, that's a great idea. We could, and the most important thing is that they had this we can do it thing, which I'm as a scientist, I'm cautious. I think about things. I want to plan ahead. I'm sort of like thinking. And they were like, it was almost like working with cowboys sometimes. Don't worry. It's going to work. Where we wanted to put our first habitat, the habitat that I lived in for several days, and I did my first, analog mission, it was basically situated somewhere on the outskirts of like an army base. Okay. So in the southern of Israel, there's like several army bases.
Yeah.
And I was like, there's no way they're going to let us put the habitat there. And my guys were like, don't worry. We talked with this guy and that guy and that guy. And I started getting these, confirmation letters from heads of, I don't know what in the army saying at this date, it's okay. We don't have any firing, drills going on, you're gonna be safe. And I think the top of it was some clergy mistake, but it was a formal letter from the Ministry of Defense telling us that they allow at these dates to build an international space station, in the Israeli Negev. And I was like, that's it, we build an international space station, that's what they think. I'm not gonna argue with that. I'm cool with that. So having a vision, that's nice, but having this go get it attitude was a big, big part. And I had terrific partners for it. I had Dr. Hillel Rubinstein, I had Roy Naor, a geologist, Hillel Rubinstein is a physicist, Roy Noor is a geologist, Alon Shikar and Moshe Tzagai were the architects. We had Yuval Porat, who was an engineer. So he was not only an engineer, like an aeronautics engineer, proper aeronautics engineer working in aeronautics, like a serious guy, but he was also he was many years a volunteer for the Austrian Space Forum.
Ah, very good.
Yeah, so we sort of had an understanding of how they would work, what would be a good experiment. And we had in our first mission, we actually had a first joint experiment with the Austrian Space Forum when they had it in Oman. And it was like a spatial situational awareness between two astronaut teams, using magnetometers to test, different stuff from the atmosphere and Mars or something like that. So it was loads of fun. We were doing something nobody else has ever done in Israel. Until now, it's like super unique. Super unique.
It sounds amazing. And you're absolutely right. You do need those people that are willing to just take action and worry about the details later. You got to have like that balance of the thinkers and the doers. I think that's super important. So let's get back to astrobiology and detecting life on Mars. So we mentioned a little bit about the microbes in rocks. I'm sure there's loads of like topics on this, but
yes.
ways of how would you go about detecting life on Mars? Like some other ways. So we've already mentioned one.
Right. So at the moment, excuse me. So it deals very heavily with what is the definition of life, right?
Very true.
Basics, you're a scientist on Earth, you found something, you pick it up. Is it alive? Is it life as you know it? As you don't know it? If you don't know it, how do you know if it's alive or not, if it's a life form?
Yes.
So I have to tell you that, first of all, we really like to debate about these things. It's an ongoing issue about, how to define life, how to define it, what kind of test, what is proper, what is not proper, what will give you like a positive, absolute result, what might not work, it's a constant discussion. This discussion has not ended at all. So everybody who feels like, joining in and telling us, how to find life forms, feel free, to contact us, the astrobiologists. We are happy to discuss it. And what we like to do, so it's a big topic. So first I'm going to say that when we think about a life form on planet Earth, today, the sort of consensus goes to that it's going to have some sort of a code, a very stable code. For us, it's the DNA. For another life form, it could be something a bit different, but it's going to be a very stable molecule. And it's not going to be such a simple molecule. It's going to have some sort of a code on it. And then you're going to have, for instance, you're going to have an envelope, because there's no way evolution would happen without putting all these different molecules in an enclosed environment, in a very closed environment. And the third thing is we're looking for metabolism, okay? We're looking for metabolism, some sort of like activation on the molecules within an envelope. So you're looking for some telltale that's telling you that something is happening there, and there's different energy levels perhaps. We have ATP, maybe other life forms have other things, but we really want that sign as well. So we have the envelope. Yeah, sorry.
If you're looking, it's probably a very simplified way of looking at it, but if you're looking for metabolism, if a species is like, if it's a life form is dormant, but it's still alive, you wouldn't be able to then detect it because you wouldn't
Exactly. If you look at a tardigrade, when it's hibernating, when it's completely void of any water molecules, and that's how it can survive, this vacuum and low temperature and radiation and whatnot, as research has shown, then he's not acting. It's full frozen hibernation. You're not going to detect anything. And that's why when scientists work these things, we don't have answers for everything. And we're really, really careful about saying, look, this is the consensus of what is life. But if I have something in an envelope and it has a really, maybe it has a population of molecules, some are short, some are big, some are complex, some are, something like that, but it's not moving or anything, you know what, that's okay. I can live with it, because I know that if I have the envelope and I have certain amount and diversity of molecules within it, that's something that, it's a really big clue. It's like a huge, huge, huge clue. So far, all the equipment we've sent to Mars, and those are mostly the NASA Fantastic rovers, what they do is that they use a spectrometer, which basically points a laser, let's say at a rock, and then vaporize like a little part of that rock. And in vaporizing it, it detects all the different atoms and the molecules that came out from that, you know, volatile area, I would say, right? So you can detect interesting molecules, and they have been saying, look, we found organic molecules. They have a lot of, you know, carbon, they have a lot of hydrogen, and so on and so forth. But just detecting a molecule is just not enough. It's just not enough. So you want to have the spectrometers, of course, but you also want to check for other types of molecules. You want to get that shape. You want to get some sort of an evidence of an envelope of a, you know, whatever cell, you know, cell structure somehow. And you would really like to see some sort of form of metabolism if you really want to nail it out. Now, there are two big problems or two challenges in this approach in having this is that, first of all, there was this really famous meteorites from Antarctica from Allen Hills, ALAH 0084. And that was a meteorite that we know came from Mars, landed on planet Earth, like many other meteorites. And in 1996, there was this Science article by David McKay, which basically shown like a blow up, like a microscopic image of a tiny part of this meteorite. And in that image, you saw something that looks a little bit like I don't know, like I would call it a cyanobacteria, but basically it looked like a little worm with like, you know, little cells inside it. Like it was a very thing. And he claimed that he wasn't sure, but he thinks that was a fossil from Mars, right? So we had a microscope. That's what we had, you know, we took it. It looks like a microscope. It looks like the thing. And then what happened ensues like one of the biggest debates ever, like in science, whatever. And eventually the consensus today that it has nothing to do with fossils and it's not alive. And you cannot say that that part has anything to do with life forms from Mars, because the fact that it looks like it, it's not enough. It's not enough. And we haven't even sent a microscope to, you know, to Mars, as far as I remember. We didn't send a microscope. So we sent a spectrometer, but we haven't really looked into how to detect life on Mars. And actually, even the Jezero rover, which have 27 cameras upon it and 3 different spectrometers, is a very impressive piece of work. Don't get me wrong. I don't know, it costs like what, $250 billion, right? And a lot of smart people, and the MOXIE, you know, thing that, you know, generates oxygen. So it's a great machine, but it's not a machine really designated, in my opinion, and other people, to really look for life. And it's the first time that they're sending some rover to actually a place that they knew probably has the best chance, as we understand it, to have life on Mars. Because the Jezero crater is not just a crater, it's actually a delta. There was a river flowing in for billions of years. It had water, there's carbonates, there's olivine, and these are generated from, you know, water environments. So NASA till now, with, you know, the planetary protection procedures and all that thing, never sent any of its rovers to someplace that even might have any evidence of life, just for the sake of not contaminating it and not destroying it, you know, making it out of balance or anything. And I have to tell you, the whole issue of getting it back to, so it's a sample return mission, this, you know, rover 2020.
Right.
And some NASA folks are really unhappy about that, And what they say, so the rover is built, it has this can, so it has this robotic arm, and it's supposed to, you know, take and put the rocks inside and close the lid. And it's supposed to stay on Mars until another mission arrives, picks it up, and brings it back to Earth.
Okay.
And the reason they did it firsthand is because through several workshops led by NASA and leading universities in the United States, they realized that they cannot make a rover that will have all these amazing tools. You know, we have so many tools today in labs to make sure that this thing is alive and this is not. And they said, there's no way that we're gonna build something like that. So it's better for us to think about a sample return mission, bringing back, of course, very safe, you know, laboratory procedures, biohazard 4, you know, everybody's throwing these fancy words that Hollywood movies then, you know, go and do fancy stuff out of it.
Yeah.
But anyway, that was the logic. They're saying, we cannot build it, let's bring it back home. But some of the scientists are saying, look, it's really dangerous. We don't want to bring even something dormant or something that was here, because we really cannot appreciate it. You know, it's about humility in a way. It's about, there's a capacity to what we know, And there's a bigger thing going on that we don't know. And just the issue, we know so much about how fragile our, biosphere is, and how much you have introducing species and endangered species, and, all these things are happening. You really don't want to bring something you don't know anything about it, even close, to our environment. So,
I can see
there's
the
a lot of
arguments from both sides, though, because there's the enormous things to learn and the curiosity, but then, like you say, the balance on what could you be introducing into our ecosystem, unless you can keep it contained somehow. I mean, you'd hope they can keep it in some kind of a contained chamber or something like that, but I don't know what their process.
So they think they can, and they, with the moon rocks, there was no, moon is like a dead thing. There's no atmosphere, nothing was ever, no water flowing ever on it. So there was no issues with the moon rocks, and still they kept it, like in this bunker and in this, incubator and all that stuff, and you can touch it with special gloves and everything. Yeah. But Mars is different, Mars is different. Mars was different from the moon. It is different now. It's a fascinating place, I have to tell you. I wish we could visit it.
Oh,
you and me on a tour.
Jump on that spaceship at some point and go to Mars. I'd love to. I mean, it's such an interesting topic. It's such an exciting topic.
Thank you.
So, aside from the Earth, obviously, and maybe Mars, where in the solar system do you think life is most likely to exist?
Speaking as a person who dealt with some extreme environments, I would put my bet on Europa and Enceladus. Enceladus, I don't know, sometimes people pronounce it a bit different. Now, Europa, one of the icy moon of Jupiter, is super icy, okay? Don't get me wrong. It's like a really cold place. It has a 15-kilometer, length or width of ice on top of it, covering it entirely. But they found out through recent research that it has a salty liquid ocean within. And what I'm saying is that if you want life forms, you need a form of energy to get, it could be a warm temperature and so on and so forth. And what we know from the Jovian moons is that you have very fierce magnetic storms that are coming from Jupiter and are affecting all its moons, actually. So I'm thinking, and when you look at the ice with Europa, and we have these orbiters looking upon it. I forgot the name of the mission that went there all of a sudden. Was it Juno? Maybe Juno.
I don't know.
I'm not sure. Skip my mind. So when you look at the ice, you can see it's actually it's a dynamic ice. There's like cracks there, and then in a second set of pictures, the cracks changes and so on and so forth. So what you see is a dynamic rotation, and you see new ice is formed and pushed to the layer. So there's some sort of a dynamic going on there. It could be from the magnetic fields, it could be from other things. Once I'm thinking there's energy in that system, and you can calculate that type of energy, you can have some theories about it. And once you can calculate it, you say, okay, I have liquid water, I have salt, so I have minerals, and seems to be that I have an energy source. So if you take into consideration 4.5 billion years, something can actually, be alive.
Yeah.
And live very happily there, very much adapted to it, In Israel, like in, caves are a great example as well. Like caves on planet Earth, you find things inside caves that took their own, 500,000 years to develop, and they don't have eyes. They're all white. There's no need for anything, because there's no light. And they're really, really happy there. They have all the nutrients. They can eat whatever they want, fungus growing underneath, some rock or something. So life adapt very well if you give it like a stable environment for, like 500,000 and 20 million and 500 million years. So I think life can be it. And Enceladus, we know there's some really interesting organics that it has plumes going into space, and even the Hubble telescope detected them. So I think it's a cold place as well, but looks like there's something happening. And if you have plumes going out, it means there's energy sources, right? Like going out and pushing this ejecta, pushing this, material out of the planet or out of the moon. So I'm looking forward for that, but really, I don't know what's going to happen. I'm pretty sure that we're going to hear a lot more exoplanets, before we actually visit them with any kind of scientific, tools.
Sure. Yeah. So getting back to analogs, what do you think are some of the biggest challenges in space exploration? I mean, there's tons of them, but how do you think analogs help address some of these challenges?
Can you give me a challenge, for instance, or what comes to your mind? I don't know. There's so
Um,
many.
I mean, there's just so many, like you say, but I guess what are the ways that analog missions help in space exploration? I know obviously there's a lot of research that goes on in like human factors, teamwork, and those kind of things, and they also test equipment and they do a lot of that. And then there's the other side where talking to like Habitat Marty and those guys, they're very much concerned with sustainability and like arid environments and growing vegetables and things in those. So I don't know, like from your perspective, how do you think the analogs might help in perhaps astrobiology research or things relevant to that?
So, so the astrobiology aspects is actually pretty fascinating. So the DMAR site is on a hyper arid area. It's considered, some part of it is like an extreme environment. And what we did in one of the last mission is that we tested, so it's not astrobiology. I'm not looking, I'm not looking at the origin of life or anything like that, but I'm doing a very interesting cross-contamination microbiology testing. So what's happening is that if you have a crew landing anywhere in the solar system, they're going to come with their own gut microbiota, and they're going to come with their skin microbiota, and their entire equipment is going to come with Earth microbiota. So you're planting a little part of Earth biosphere on a new planetary body. Now, one of the things that we tested in this scenario is that we said, okay, we are aware of the situation. There's going to be forward and backwards contaminations. How can we, have a grid in space and time to actually catch up what's going on? Who's taking things out from the habitat? And, they're going for EVAs, they're using the rovers, they're doing stuff. So they're going away from the habitat, and they're for sure, shedding some skins, not skin cells, but shedding, things from the equipment surface and so on and so forth. And we know from Earth that we have a lot of resistant bacteria, like really tough bacteria that can, it can't survive on Mars. Bacillus and spores and things like that, they're excellent for a Martian atmosphere, nice afternoon. So what we did is that we decided that we're going to have like a few spots around the habitat, which we were fairly certain will get forward contaminated. And then we picked a site which was never visited before or after the mission, sort of like not sterile, of course, because it's a hyper arid environment. It's a natural environment. And we sampled them every 2 days. And we did like what we call now 16 bacterial sequencing to see the microbial diversity. And what we checked is also the analog astronauts' gloves, because we wanted to see you use your hands a lot. Humans use their hands a lot. So you take a new pair of gloves. In our case, it was like nitrile gloves that you put on top of the big, suit gloves. And we swipe them before an EVA and after an EVA, right? An extravehicular activity. So basically the idea was to detect what their hands, even if they didn't dig anything, even if they didn't do something really with the land or something, we tested what their hands, brought back with the gloves, with swabbing of the gloves. And what we're looking for is things coming from outside, going inside the habitat. So it was just, like a preliminary study and everything, and it's going to be published hopefully very soon, crossing fingers. But this is what we like to do, like with planetary mission and in analog missions, which we don't see enough of it. So you see the technology testing, you see the human psychology and monitoring, like seriously, as chief scientist of DMARS, 50% of the science proposal that we get, and we get plenty, is about psychology.
Right.
It's a lot, actually. 50% of the science proposals is for some aspects of the human spirit, the human mind, the human psychology, how it will adapt into this isolated environment. So in astrobiology and microbiology, there's a lot more to do.
Yeah.
And it's a situation that NASA has not really tested, because NASA always says we have astronauts on the International Space Station. They're not going out to any planetary missions. There's no point in us monitoring what's going out and what's coming back, because we just need to monitoring, what's happening inside. And they do a fantastic job at, hygiene and making sure that this type of fungi and this type of bacteria, they don't get, overload in the system. an important aspect in an isolated mission. But they never do the other thing, which is let's go outside, and the pattern of search when you go out and you want to collect rocks or anything like that. So, and test that. So this is a demonstration study, for instance, what we can do in analog mission with the human and the microbiology side of it. That's what I can offer.
Yeah. We're looking to the future, I mean, currently NASA's staying in the ISS, but what about Mars? That's where we're all excited to head to. So that's where.
Do you think we're going to have first like human on Mars or humans on Moon? What do you think?
Well, I think so. They're headed to the moon again. So I think that's something obviously that they're trying to do, is build this little base on the moon. So I think that will come first. But
what is your guess? Like from a year point of view?
It's going to sound, Mars, I think, is beyond a year. I don't think, obviously, I think even in the next decade, Mars is unlikely. I know there's a lot of very optimistic talk of getting to Mars within the next decade, but I actually think realistically, that's very, very difficult. I mean, there's all kinds of difficult concerns, like, but back to the human aspect of it, the effect of microgravity on the body, human body, there's challenges, they're still there. So, I'd love to believe that we'll be there. Certainly in my lifetime, I think we should be there. But I think maybe not the next decade, maybe beyond that. And God, I'd love to go if we do get the chance. And like you said as well, it sounds like you'd also love to take that trip to Mars. I mean,
for sure.
For
sure. I just can't wait for some of the numbers of sending a human to anywhere outside of Earth to get a little bit down. I just can't afford it. Anyway, I look at it, it's like, do I need to sell a kidney or something? Like, just even Virgin Galactic, which a lot of people have bought a ticket in Virgin Galactic. A lot of people bought a ticket with Virgin Galactic, and I remember Virgin Galactic starting 7 years ago already, something like that. Pretty long.
And it always feels like they're saying, "Oh, we're nearly there. We're just on the cusp of it. A few more years." You're the first out there, and I mean, it sounds kind of affordable. We're talking about hundreds of thousands. We're not talking about I don't know,
millions.
But even so, like you say, there's still a lot of money. So bring that price down so that
I say, let's do it together. Talk to Tom Cruise. Say, look, you're doing an important service for the humankind. Take us with you to the ISS. Don't worry, we'll manage. We're analog astronauts. We manage.
Absolutely. We should be next. So do you have a funny story from your analog missions that you can share with us? I'm sure you have loads, but thinking of one off the top of your head.
Oh my God. Like a funny. What was funny? I don't know. Everything was unbelievably, it was surreal what was happening on the mission. I'm trying to think of a funny thing. No, funny is not the word that comes to mind, I had so many, it was such a rich and complex experience for me that, I don't think I really wrote it down actually properly to nail down to too much happening. I remember, so we were in this habitat. Our habitat is about almost 60 square meters. That's pretty awesome.
Yeah.
And what we did is that we had one part of it had the bunks, which were beautiful, wooden, how do you say this? Wooden cabins?
Yeah.
Not cabins, more
rooms?
No rooms, just
niches? Bunks?
Bunks made out of wood, though. It was very pretty. And then on the other side, we had my lab, which was not really a fully fledged lab, but still I had a lot of experiments to run, 3D printing, And the architect, who is a terrific, funny, creative, amazing guy, comes to me on the second day, at the end of the second day, we're just hanging, just understanding all the different things. He goes I smell something. And I'm like, what are you smelling? And he says I don't know, but it feels like a chemical. Could you check your bottles and Now, I work in labs all the time. I don't smell anything at all by this stage. Everything is I know what, we microbiologists are we're really exposed to a lot of different, not so good smells. So I didn't know what I was talking, and I'm looking at him and I realized that there's this gap between us because I'm a scientist and I'm experienced scientist. So I'm used to the smell, and I know to differentiate between good smells and bad smells in my profession. and he was smelling a little bit of like a 5% acid. Like he was so sensitive, but
Yeah.
because he had no knowledge of science and he never ever, you know, dealt with that thing, with that area, then every little thing for him was basically could be a threat.
Yeah.
And I was like, okay, dude, tomorrow you're going to join me for 1 hour in my lab and I'm going to show you everything, you know, sort of like to make him a bit more comfortable
Yeah.
and tell him this is good, this is good. And, you know, I was super careful in all my experiments, in all of DMAR's experiments, is that because we're in a nature reserve, right? And because of the values that we hold about sustainability and everything, I never allow any experiment which has, you know, any kind of, you know, harsh material or a bad chemical to work within the habitat and within the mission. It's a big no-no. So the worst is like, you know, ethanol, 70%.
Okay.
You know, the worst.
No big deal.
So I had to show them that, and then it was like, and you know, since then we were like buddies forever, so on. But yeah.
Right. So what's next for you in terms of space? I guess continuing with your research.
I am continuing with my research. I'm also doing science education. So I'm part of I'm the current manager of a science education program called the Young Astronauts, supported by the Israeli Space Agency. And I've been involved with this program for like 3 years now, and it's been quite amazing. So we train high school students as astronauts, but we teach them on the way a lot of space sciences. So we talk with them about, you know, remote medicine and space architect, like things, not just astronomy and astrophysics, but, you know, astrobiology.
Yeah.
And on the way, we train them as a team to do eventually an analog mission.
That sounds amazing.
It is. I don't understand how nobody else is doing that.
Yeah.
Come on, people. You have analog habitats. Do something, you know? And it's a brilliant program because working with high school students is a bit, you know, you're not always, the material is still, you know, happening and evolving, to say, and you have to take that into consideration. But in Israel, we do, you know, there's compulsory army service. So basically all the children, after they finish high school, they don't go to college and they don't go to university, but most of them, they go to this, you know, compulsory army service, 3 years for the boys and 2 years for the women. So everybody in Israel already knows that they're going into this, you know, scene, and they talk about it, and it's okay. It's very normal for us. So for them to do an analog mission is actually a great way for them, you know, to look forward in their life and what they're going to do afterwards, after high school. It's like a sort of a break, and it gives them the taste of space exploration in a way that nothing else. You know, you can, you know, meet them with astronauts, and you can talk with them, and you can show them NASA educational resources, which are awesome. However, doing an analog mission, training as a team, understanding the geology around you, what does it mean to be a planetary scientist? How do you follow the water? You know, how do you navigate?
Yeah.
How do you navigate? It's not easy for high school students, any of these topics. And I'm very happy to be involved with that one. So I'm trying to continue my research and also do the science education thing, mainly.
Is there anything else that you'd like to share with us today, and how can people follow you in your work?
Yeah, sure. I mean, I'm very accessible via the internet at the moment. You know, feel free to drop me a note with LinkedIn or Facebook. I'm not big on Instagram, even my Twitter. I do have a Twitter, but I don't really go a lot into that. You know, for people who are seeing this interview, we have a life outside the computer. We really like our life outside the computer. I don't spend my entire day doing this all the time. I just have other things to do. I'm sorry. I understand that's your connection to me, but please understand that. So you can contact me. You know, there's terrific websites for DMARS, the, you know, Mars Society website and Facebook. Again, me and LinkedIn, me in everywhere else. And yeah, I'll be happy to hear your thoughts. And if you have any interesting projects, you need another astrobiologist eye on it, I'll be happy to be around. Sure.
Yeah, and I'll put links in the description for people.
And we both hope that within our timeline, right, we will get to space. We will work
100%.
and live in space.
Yes, definitely. And if anyone's interested in learning more about astrobiology, I highly encourage you to check out Reut's online. There's an online course she did recently, and it's on YouTube. And
With Astronera.
yeah,
With
very, very good.
Astronera.
And I'll put that
Amazing.
link to that as well in the description so you guys can check it out. So Reut, thank you so much for talking to me today. It's been amazing.
Thank you, Jess. Go on. Let me know what your next projects are. I'd be happy to check them out. Amazing work. Thank you.
Thank you. Thanks so much.